Assalamu alaikum, dear students. Welcome to the course Principle in Animal Life 2. This is lecture number 12. The topic is Exceptions of Mendelian Genetics. Discussing agenda for the topic will be inheritance patterns are often more complex than predicted by simple Mendelian genetics. The relationship between dominance and phenotype, frequency of dominant alleles, multiple alleles, and Pleuotropy. Lecture outcome. After watching and listening this lecture, students will know about different crosses that violates typical Mendelian principles of genetics. Introduction. Inheritance patterns are often more complex than predicted by simple Mendelian genetics. We have already studied about the Mendelian genetics, the two laws formulated by Mendel in light of his findings, and these laws are predominantly observed throughout the living kingdom. However, we do have certain cases where the crosses between the genetic crosses does not obey the rules and principles of typical Mendelian genetics. So these crosses which does not obey Mendelian genetics are known as exceptions of Mendelian genetics. In the 20th century, geneticists extended Mendelian principles not only to diverse organisms but also to patterns of inheritance more complex than those described by Mendel. So Mendel worked with the pea plant in 1860s, and thereafter when in 1901 Mendel's work was rediscovered, the people belonging to other areas of genetics, working with animals and working with other plants, they also started observing the genetic crosses, and then they tried to explain the results in light of the two laws of Mendel, and they found some exceptional cases where the outcome of genetic crosses were not obeying the principles of the Mendelian genetics. For the work that led to his two laws of inheritance, Mendel chose pea plant characters. They turn out to have a relatively simple genetic basis. Each character is determined by one gene for which there are only two alleles, one completely dominant and the other completely recessive. There is one exception. Mendel's spot shape character is actually determined by two genes. So Mendel was lucky in a way that he studied only the pea plants, and the pea plant has various characters, and the characters which Mendel studied were controlled by a single gene, and this single gene had only two variants. The characters which Mendel studied had only two variations, but this is not the case throughout the living kingdom. There are variations, there are genes which show up in multiple copies in a population. There are instances where the F1 hybrids does not resemble either parent. There are instances where a single gene controls more than one character. There are instances where a single character is controlled by more than one gene. So all these conditions, they disobey, they violate the principles of typical Mendelian genetics. Not all heritable characters are determined so simply, just like the Mendel observed when he found F1 hybrids exhibiting the features of the dominant parent, and then the F2 generation where the recessive parent also appeared. So the things are not so simple everywhere. Mendel himself realized that he could not explain the more complicated patterns he observed in crosses involving other P characters or other plant species. Mendel himself came across some instances or crosses where even his own laws were not sufficient to explain the outcome of these crosses, but then Mendel didn't describe them or didn't documented them because he thought that if I document something which I am unable to explain completely, will obviously decreases the importance of my work. This does not diminish the utility of Mendelian genetics, but still the Mendelian rules and the laws are very much common, and they are observed in a lot of cases across the living kingdom. However, because the basic principles of segregation and independent assortment apply even to more complex patterns of inheritance. So although the rules and the principles of genetics elaborated by Mendel courtesy of his experiments are not applicable 100% in every case, but still the inheritance of characters do govern these basic rules. So here in this section we will discuss the genetic crosses which does not obey the rules of the typical Mendelian genetics, or which does not obey the two laws of Mendel. Now here we will discuss such crosses which does not obey typical Mendelian genetics and which are controlled by only a single gene. Remember that some traits are polygenic in nature. They are controlled by more than one genes, like skin color. So in our next lecture we will discuss the exceptions of Mendelian genetics in polygenic traits. In this lecture we will only discuss the exceptions of Mendelian genetics, keeping our focus on only those characters which are controlled by a single gene. Now, alleles are the alternative form of a gene. Alleles can show different degrees of dominance and recessiveness in relation to each other. So in Mendel's case, there were two distinct forms. One was completely dominant and the other was recessive. There was no intermediate form. However, the alleles can show different degrees, different levels of dominance and recessiveness, as we will discover in the next few slides. In Mendel's classic P crosses, the F1 offsprings always looked like one of the two parental varieties. So if you recall the Mendel's first law, then the F1 hybrids only showed the phenotype of the dominant parent. So this was complete dominance. Therefore, the Mendel law of segregation, or the law of segregation, is also known as the law of dominance, where one allele was completely dominant over the other, and the F1 hybrids they exhibited the phenotype of the dominant parent. In such situations, the phenotype of the heterozygote and the dominant homozygote are indistinguishable. Dominant homozygote means when the genotype of the F1 hybrid for a particular trait is homozygous. So here, if only one dominant allele is present, then still the phenotype is controlled by the dominant allele, and if two are present, then still they control the phenotype. So the homozygous dominant or heterozygous dominant both expresses the same phenotype. For some genes, however, neither allele is completely dominant, and the F1 hybrids have a phenotype somewhere between those of the two parental varieties. On the right side of your screen, you can see the example when a plant having red flowers are crossed with another plant having white flowers. Now the F1 hybrids will exhibit pink color. Now you can see here, pink color resembles neither of the two parents, and it is somewhat a mix of either parent. So we can say that the F1 hybrids are exhibiting the phenotype of both parents to a certain degree. So when Karl Correns, one of the rediscoverers of Mendel's work, was the first person who observed this phenomena, and when he reported this phenomena, a lot of people at that time started thinking that perhaps the blended hypothesis is correct, because the F1 hybrids are exhibiting features of both the parents. But when the F1 hybrids were crossed with one another, the F2 generation gave a typical Mendelian ratio where three of the plants were having color and one of them was colorless. So it was a typical Mendelian ratio, 3:1. Now this phenomena is called incomplete dominance, where neither of the parent seems to be dominant, seems to be controlling the phenotype of the F1 hybrids. And this example is seen in snapdragon plants. All the F1 hybrids have pink colors. So this third intermediate phenotype results from flowers of heterozygotes having less red pigment than the red homozygotes. So here you can see that the F1 hybrids where the one allele each from either parent is present, and the allele determining the red color is not dominant enough to completely control the character, and the allele controlling the white flower color is also expressed to a certain extent. And in this case, where both the alleles are in homozygous form, then the color of one of the parents is exhibited. Another variation on dominance relationship between alleles is called codominance. In this variation, the two alleles each affect the phenotype in separate, distinguishable ways. So this is yet another case where we don't see complete dominance. Now, to illustrate the relationship between dominance and phenotype, we can use one of the characters Mendel studied, round versus wrinkled pea seed shape. Now, why we called an allele dominant and why we call an allele recessive? What is the significance for that particular terminology? How an allele which is termed as dominant controls the phenotype to a different degree as compared to the allele which is designated as recessive. So the dominant allele which is having the seeds which are round, so the allele determining the round seed shape is termed as dominant, whereas the allele which determines the wrinkled shape is termed as recessive. So what actually happens at the molecular level? The dominant allele codes for an enzyme that helps convert an unbranched form of starch to a branched form in the seed. The recessive allele, which determines the wrinkle shape, it codes for a defective form of this enzyme, and therefore it is unable to convert the unbranched form of starch to branched. Therefore the unbranched starch accumulates in the seed, which causes excess water to enter the seed by osmosis. And later, when the seeds that have absorbed excess water dries, they become wrinkle shaped. If a dominant allele is present, no excess water enters the seed because the unbranched starch is converted into branched form, and the seed does not absorb water. One dominant allele results in enough of the enzyme to synthesize adequate amount of branched starch, which means that dominant homozygotes and heterozygotes have the same phenotype, that is round seeds. So the F1 hybrids, whether they are homozygous or whether they are heterozygous, they will exhibit the round seed shape because only one allele is enough to control the trait. A closer look at the relationship between dominance and phenotype reveals an intriguing fact. For any character, the observed dominant recessive relationship of alleles depends on the level at which we examine phenotype. Now, we have now seen that the relative effect of two alleles range from complete dominance to incomplete dominance of either allele to codominance of both alleles. So it is important to understand that an allele is called dominant because it is seen in the phenotype and not because it is controlling or it is suppressing the effect of the recessive allele. Both the alleles are independent to express, to exhibit their potential. So alleles are simply variations in a gene's nucleotide sequence, as we studied that in our previous lectures, and both the alleles, they exhibit themselves according to the potential they have. So thereafter, the dominant allele, which synthesizes more proteins, becomes the controlling factor that controls the phenotype. So when a dominant allele coexists with a recessive allele in a heterozygote, they do not actually interact at all. So there is no interaction between the dominant allele and the recessive allele. That is explanation of typical Mendelian genetics. In our next lectures, we will study examples where the alleles at one locus interacts with allele at other loci. So it is in the pathway from genotype to phenotype that dominance and recessiveness come into play, just like we observed in the round seeds and the wrinkled seeds. Now the allele determining the round seed doesn't suppress or control the expression of the allele determining the wrinkled shape. It is actually this dominance and recessiveness is determined in the pathway during which these genes express themselves. So now we discuss multiple alleles. Multiple alleles means that in a single individual, a gene can exist in a maximum of two variant forms, which are called alleles. But in a population, there can be more than two alleles for a specific trait, which will determine the phenotype to varying degrees. So only two alleles exist for the P character that Mendel studied, but most genes exist in more than two allelic forms. Now, for example, the blood groups. So an individual can have a maximum of two alleles, which determines the blood type, but in a population there can be different forms of these alleles which determines the blood groups. So the ABA blood group is in humans, for instance, are determined by that person's two alleles of the blood group gene. There are three possible alleles. A person's blood group may be one of four types: A, B, AB, or O. So you can see here there are three different alleles in a population which determines the blood group. But a single individual can have a maximum of two alleles because a gene can only exist in two alternative forms, but in a population there can be more than two alleles for a given locus. So this is yet again a violation of typical Mendelian genetics, where we studied only two forms of a particular gene or a particular phenotype: tall, dwarf, flower position, axial, terminal, seed shape, round, wrinkled, flower color, seed color. So there were at a maximum of only two variants, but in case of multiple alleles there can be several variant forms of a phenotype. These letters refer to two carbohydrates A and B. They may be found attached to specific cell surface molecules on red blood cells. An individual's blood cell may have carbohydrate A, then its blood group becomes A, or carbohydrate B, then its blood groups become B, or having both A and B, then its blood groups become AB, or neither of the two carbohydrates, then its blood group becomes O. So matching compatible groups is critical for safe blood transfusion. Of course, when you are donating blood to someone, the compatibility of the blood groups is important. You can see here there are 3 alleles for determining the blood group in a population. So a person's RBCs have carbohydrate A, the blood group is A. Having carbohydrate B, the blood group is B. Having carbohydrate AB, the blood group is AB. Having none of the carbohydrates AB, the blood group is O. And these are the appearances of the RBCs. So you can see here this person's blood group is A, where both the alleles are AA, or one of the allele is A. Here the person's blood group is B, where it is homozygous. It is heterozygous, but still the blood group is B. Now here both A and B alleles are present, so the person's blood group is AB. And here neither of the carbohydrate A or carbohydrate B is present on the blood cells, so the blood group is O. So this is yet another example of the exceptions of Mendelian genetics. Now, final topic for this lecture is Pleuotropy. So far we have treated Mendelian inheritance as though each gene affects only one phenotypic character. So if you go back to the Mendelian genetics, each gene controlled only one character. Some genes controlled height, some controlled seed shape, some controlled seed color, some controlled flower color, some controlled the position of the flower, but one gene was unable to control more than one character. So here in the case of Pleuotropy, which is also a violation of Mendelian genetics, a single gene controls more than one character. Most genes, however, have multiple phenotypic effects, and this is a property which is called Pleiotropy. Pleiotropy is a phenomena when a single gene controls more than one character. In humans, for example, pleiotropic alleles are responsible for the multiple symptoms associated with certain hereditary diseases such as cystic fibrosis and sickle cell disease. So, in the case of genetic disorders, only a single gene is affected in cystic fibrosis, where a sticky substance accumulates in the alveoli of the lungs, and therefore it is difficult for the alveoli to inflate. And then is the sickle cell anemia, where the shape of the RBCs becomes sickle-like, and it becomes fragile and unable to transport oxygen. So these characters are controlled by a single gene, but once mutations occur in that single gene, then the person observes a whole lot of symptoms. We can say that the different symptoms which are being shown by the patient, in fact all these symptoms were prevented when the genes was in its normal form, in its wild form. But once the gene mutated, then it lost its control over the different features it controlled, and therefore the different features then resurfaced in the form of different symptoms. In the garden pea, the gene that determines flower color also affect the color. So here we also have an example of Pleuotropy from the Mendel's pea plant, where a single gene is controlling two characters. So given the intricate molecular and cellular interactions responsible for an organism's development and physiology, it isn't surprising that a single gene can affect a number of characters. So these biochemical pathways are indeed very fascinating, and you can observe genes which have role in several biochemical pathways. So indeed the phenomena of Pleuotropy is quite often and widely seen in the living kingdom. And remember, this is the violation of typical Mendelian genetics. In our next lecture we will discuss few more examples of the instances where the typical Mendelian genetics or the rules are violated. Thank you very much for your time. Allah Hafiz.